and I have a case.
Okay,
if that's okay with you.
Let's run the gauntlet, bro.
Perfect.
We're going to be live in YouTube in 2 seconds. Starting the webinar. 5, 4, 3. You
want me to broadcast?
Yep.
Okay. There you go. We'll just give everybody a minute or two to join. Okay, let's get started with this week. I'm very excited about our team today. I just want to welcome everybody to the Miami Global Brain Tumor Symposium. Just a couple introduction slides before we get started. My name is Michael Ivan. I'm one of the neurosurgeons here specializing in brain tumor and skull base at the University of Miami, and also the co-director of our research program. And I'm joined by my co-directors, Dr. Morcos, professor and co-chairman of our University of Miami neurosurgery program, as well as director of cerebrovascular and skull base. Dr. Komatar is the professor and program director, also director of the UMBTI and surgical neuro-oncology, and Dr. Benjamin, assistant professor and director of our skull base Keynes Laboratory and brain tumor and skull base specialist. Each week we put on these sessions. This is our 27th week since COVID started. We have over 7,000 people registered for these talks, and many, many thousands of people who have watched them over the months. And I thank everybody who's done that. But I most importantly want to thank all of our administrators who have helped us out with running these sessions with the support from the University of Miami, Sylvester Comprehensive Cancer Center, and the Department of Neurosurgery, Christina, Roberto, Ingrid, Damari, and Ignacio. Thank you all so much for all that you do. For anybody who's joining tonight, if you have any questions or want more information about the University of Miami Neurosurgery, you can always follow us on social media. We're on Twitter, YouTube, Instagram, and Facebook. Also, you can find more information about all of our Symposiums on our website. And that brings us to the upcoming Symposiums. So tomorrow, every Thursday, Dr. Morcos leads our skull base and cerebrovascular symposium. We have a great set of panelists tomorrow: Dr. Gentili, Dr. Cavallo, and team are going to be joining us talking about endoscopic approaches to giant pituitaries and anterior skull-base meningiomas. So please join tomorrow, 5 o'clock. And also next Monday is our first pediatric neurosurgery symposium, looking at pediatric neurosurgery oncology. We'll be starting at 5 o'clock on the 9th of November. So please be sure to join us for that. And a quick sneak peek into next Wednesday's Miami Global Brain Tumor Symposium. Dr. Golfinos from NYU, he's the chairman of neurosurgery there, will be joining us to give us an update on the surgical treatment of NF2. So please be sure to join us next week. Some housekeeping before we begin. Please, we try to make this as interactive as possible. So please use the Q&A button at the bottom of your screen to ask your questions. We'll ask Dr. Shigeru at the end of his talk and then in between the different cases, his opinion and try to get to all your questions. We don't offer CME for this program, but you do get an email confirmation documenting your participation. And please be sure to like, follow, and share all of our videos so that we continue to grow on Instagram, Twitter, and YouTube and share the fantastic knowledge of all of our speakers with more people. So getting to this week, we have a great set of panelists. Dr. Hamp, again, joining us back from Westchester Medical Center. He's assistant professor, also alumni of Columbia, is specialist in minimally invasive surgery and the section chief of neurosurgical oncology. I think you guys all know me very well. And then very special tonight, we have Dr. Eichberg, who's one of our own University of Miami neurosurgery residents. He completed our clinical neurosurgery oncology fellowship, as well as spent time down with Dr. Shigeru and Dr. Teo for international fellowship. And now he's in the laboratory doing NIH T32 research fellowship, working with myself and Dr. Ayad on glioblastoma. So Dr. Eichberg, thanks so much for joining us. Tonight we have a very special guest, Dr. Shigeru. He's joining us all the way from Australia, early morning on Thursday. I cannot say thank you enough for joining him. Mike, I've known him for more than 14 years. We go back to doing our residency together at San Francisco, where he was one of my chiefs at the General, and I can't say enough good things about him. Mike grew up in a small town in Oklahoma. He went to Columbia, graduated first in his class, and then went to UCSF. He was mentored by one of my mentors as well, Dr. Andy Parsa, who really influenced both of us and really challenged Mike kind of do some amazing work while he was in residency that he continued thereafter. Mike then went to do a fellowship on minimally invasive surgery with Dr. Teo in Australia before joining University of Oklahoma as their director of brain tumors, where he kind of exploded their program by doing over 3,000 brain tumor cases in the university in a very short period of time, and was a great director there. Mike then continued to kind of grow and develop his research abilities and went back to work with Dr. Teo, as well as then to start a new company called Omniscient, which we'll be talking about tonight. You could see here some of the fantastic work that Mike has done in the books that he's written, the glioma book, which is just a phenomenal book, detailing all of the specific eloquent steps needed for successful glioma surgery, minimally invasive surgery, management of inoperable gliomas. All are fantastic reads, and I encourage you to do that, as well as some of his other 200 articles that he's done, including a recent entire journal, monthly journal for neurosurgery, documenting all of his connectomes for glioma surgery. Mike, thank you so much for joining us, and we're really excited to hear about Connectomics and what you're working on in Australia. And we're also here at University of Miami extremely excited to be a part of working together with you to kind of implement this in our own clinical practice.
Well, thanks for having me. Basically, here in Australia, we're completely opposite to you guys. It's morning, and we're going into summer instead of winter. But what I want to talk about is connectomics. And for those of you familiar with genomics, connectomics is another omics. And it basically has profound implications. When you understand what it means, profound implications for how we think about the brain. Because what it really comes down to is we have to sit down and think about what we actually do when we remove a glioma. What does it actually mean to remove a brain tumor that is infiltrative, meaning that it can have functional tissue running through the brain? So a lot of us, and myself included, And the way we were basically taught, no one ever said this, but this is kind of what when you observe your mentors, what they were doing and how we thought about the problem, was that there was eloquent areas and non-eloquent areas. In non-eloquent areas, as long as you didn't get to an eloquent area, you were pretty well safe. You could do what you needed to do. And the main issue is just to find the eloquent areas and not go through them. And ultimately, you need to stay within the tumor, which is a really kind of a complicated concept when you realize that we know that there can be functional brain tissue running through at least the periphery and sometimes the center of some of these tumors. And the main goal was just don't cut the corticospinal tract. Probably be good if you didn't cut the arcuate fasciculus. Everything else, the optic radiations seemed to be important. All the rest of the stuff, we didn't think about it that much. And part of it was we didn't know what was really these things were doing, what they were connecting, and you couldn't find it anyway. It was too complicated. And so there really wasn't a whole lot of reason to spend much more time than don't cut the corticospinal tract. It's obviously a good piece of advice, so we didn't do that. One of the things that, since I was a very junior resident at UCSF, and we would do these very aggressive glioma surgeries, and the patients would walk out of the hospital and seem to be okay, and we'd all high five and feel good about it. But then if you've been the junior resident taking call and the patients call in, they'd often call at some odd hour when you were in the middle of placing EVDs and saving lives, telling you about that their family member had some bizarre cognitive symptom. They were making bad decisions, they were acting strange, and you would realize this wasn't hydrocephalus or a CSF leak or something you need to be worried about. It was just a feature of the fact that you removed this tumor. And ultimately, what has always fascinated me since that moment, and still has been a mission of mine, is what is the cognitive morbidity? What are we doing wrong, and how do we not do it wrong anymore? Because even if you go past these kind of subtle higher things, meaning the sublime goal of saving someone's cognition while it's taking out their brain tumor, we still have deficits that we don't understand, and we attribute to swelling, and sometimes they get better, or an SMA. But until we really have a framework for thinking about this, we're going to keep having this happen. And if we want to improve, the same as we improved with skull-based surgery, we need to better understand what we're doing, what we're treating, what our goals are, and to iteratively improve on our techniques. Because realistically, some patients are going to have neurologic deficits. This is glioblastoma is a bad disease, and we don't have a solution for it. And glioma is a bad disease, and we don't have a perfect solution for that either, low grades. But what we want to do is not have things that happen that we had no idea what we did wrong. And one of the things that we have to have a real good thought of when we think about the term of oncofunctional balance is we understand the onco part reasonably well. We don't know where all the tumor is, but we know most of it is in the radiographic anomaly. And again, if we take a margin, if possible, like a supramaximal resection, we can get a reasonable resection and change the natural history of the disease. We understand the onco pretty well, but we don't understand the function very well. We don't understand how the brain does a lot of this stuff. And whether we like it or not, we're cutting at least next to and possibly through brain networks, depending on how they're running through the tumor. Now, one of the things that we have to question is the concept of eloquence. What does that word even mean? And of course, the brain didn't make parts of the brain, like the frontal lobe, because it wanted to, absorb CSF. It did it for a reason. So what we had to think about is a different way to think about this, which is not eloquent versus non-eloquent. And when people have gone the other direction, say, well, it's all eloquent, it all does something. Well, we know that that's not really true in the way that we mean. So eloquent means if we cut it, there's a deficit. And we know that in some patients, you can do pretty dramatic things and basically leave them normal. They can go on to live a normal life. So what it really comes down to is the problem here is a poor understanding of the redundancy of the brain, what's tolerated, what's not tolerated. And the reason this is the case is that gross anatomy, eyeballing, and this is how I do it is not adequate to handle a problem this complicated. So even if you don't know it's there, it's there. So your ignorance doesn't actually protect the patient. So walking through what we mean by taking out a glioma, what are we thinking about doing? And how can we do this better? Okay, so this is a large insular glioma. And our goal is to get a resection like that on the right or some variation on that, where we do a very good removal and the patient walks out of the hospital and goes about their life like this didn't happen with the minimum amount of morbidity. But again, how do we know what the exact network anatomy is? Because the insula isn't there sucking CSF. It does something. And in fact, it does quite a bit of things. how do we know whether this patient is living without that insula because the tumor, it's rewired in some way or reorganized, versus one where this is going to be a trade-off between what the insula is doing and their quality of life. And maybe it's a trade-off that we still make to take the tumor out, but we should be doing that based on data, not based on, I hope this goes okay, which is what we do now. Now, DTI and resting state and these kind of methodologies are not new. Everyone here probably has heard of this stuff. It's been around since the 80s in the case of DTI and the early 90s in the case of fMRI. But when you look at one of these things, even myself, and I've looked at, I would say, close to 10,000 DTIs over the course of my life, doing various studies and treating patients and various things. It's really hard to know what all these fibers are doing. You can kind of recognize some patterns, you know, the corticospinal tract somewhere in that blue mess. But what are all these wires doing? And when we start talking about cognition, this is often multiple areas working together for brief periods of time to make a higher function like math or cognition, you know, abstract thought, working memory happen. So how can we possibly, with the human brain, understand the human brain? Because if the human brain was easy to understand, we'd be too simple to understand it. Well, when we start tackling a very high dimensional problem, which is what the brain anatomy is, there's lots and lots of variables here. One of the first steps in any complex data analysis, like machine learning, is to dimensionality reduce, cut it down to a number of dimensions that's tractable. And everyone here knows at least one dimensionality reduction technique. Now, there's some that are mathematical, like PCA or something, but actually one of the ones that we're all familiar with is parsolation. So you'll know a parsolation, it's called Brodmann's areas, and Brodmann parsolated the brain, meaning he divided the cortex into parcels that he determined were distinct from other parcels based, in his case, on cytoarchitecture. But you don't have to parsolate based on cytoarchitecture. We know that Brodmann's data was pretty good for 1908, but there's a lot of ways to do better. This diagram shows the HCP scheme, which did this based on functional connectivity and different task analysis as well as cortical thickness. What parsolating does is it gets the brain down to a finite number of dimensions that humans can understand and tackle, and it does it in a way that you can safely assume that all the parts of, say, area 44 or Broca's area are probably doing something similar to, you know, they're relatively uniform. And we know that the brain roughly works this way. When you parsolate the brain, there's a lot of things you can do with it. First thing, you can start to make inferences on what all these wires are doing, and I'll show some of that, but also you can take something like fMRI, like resting state, extract the bold signal at every place, compare them, and see how similar they are through things like correlation. And there's no real way to do this if you look at every single voxel on an MRI separately. And so this allows us to basically describe the brain in a way that we can all talk about something and call an area of the brain the same thing. I want to draw attention to this. This was published in Nature in 2016 by the Human Connectome Project. It's a very, very rigorous paper. If you read it all end to end, it's about 500 pages long. And there's a couple of things I want to point out. So if you look at the two brains at the top, the left is a flattened brain where they've expanded it out. And the right is the folded version of this brain. And notice that every single little boundary is describing an area that's functionally distinct from its neighbors. And they vary in sizes. But what's more important is there's some things that really show the inadequacy of how we've described parts of the brain. We describe them in terms of gyri, which of course has a big problem in the sense that everyone has different patterns of gyral and sulcal patterns. But I think a bigger one is there's areas underneath the operculum that do something totally different than the outer surface of the opercular cortex. There's areas like this area RSC, which sit inside the callosal sulcus that we don't really pay a lot of attention to. There's areas like area 46, which crosses onto two different gyri. And there's areas like 55B, which is an incredibly important area. It's a unique non-Broca speech and language area that sit inside of a sulcus that we never saw that. We didn't know it was there. And so all of these things show that how, by really thinking about things in terms of the visible anatomy, we've held back our ability to actually make progress. The brain does not care what you call a gyrus or a sulcus. It just puts the function on the cortex wherever it needs to do that. And so we need to really start to understand and talk about the brain in the way that it really is and not in the way that's simple for us to do. And so when I read this paper, I was very influenced by it. And as Mike mentioned, we started doing anatomy, because this is one thing that I found when I got into the field of connectomics. There's some really, really smart people. Many of them have physics backgrounds doing very complex math to understand the brain, but very few of them really care exactly where that area was. But we're neurosurgeons, we do care where that area is. So we wrote anatomy of this. And without going into all of it, I'll touch on some things that we've learned from the connectome. But I think one of the things that came out of it when I first started showing it to people is they all said, well, this is impressive work, but there's no way I can learn all this, and what are we going to do with this? It's too much information. So I started working on that problem because I'd like to take critical feedback and do something with it. And the first question is, how do we find those areas in a brain of a patient that we're treating? Now, in a normal person, in the research world, there's a technique called atlasing. So atlasing means that you take a list of regions, and these are the different parsolations of HCP, and you warp it onto the brain. You shift it and warp it till it fits the brain that you're on. And this is pretty well. You can see that it missed a spot here, and it missed a sulcus here, kind of. It does pretty well with this, but what about that? And one of the things I found out when I first got in the field very quickly was if you put a brain tumor patient through the open source software that people do research with, it just crashes because they make assumptions about the shape of the brain. So what I want to show is a video of what we did to help crack this. And this is a technique called reparcelation. We developed this over 2 years ago, and we've spent a lot of time working with it. And it's designed to tackle this problem. So if you take the brain, and what we want to do is assign areas. We don't understand parsolations because they're going to tell us about function. So let's say that you're looking at Broca's area. Well, we know that it's going to be about there, but it's going to be different a little bit in everybody. So what we did was we took the machine learning knows the DTI, and it looks at every voxel one at a time to see who are they connected to. The machine learning knows that that's a voxel of 44, and this one is not, because one talks to Wernicke's area and the other one does not. And it looks around there, so even into voxels that are not part of the initial assignment, and says, maybe I shouldn't put that one into the boundary. And when it finishes doing that, you now have a parsolation for 44 that actually is specific. Now, in a tumor case, the initial parsolation assignment will be in the tumor. So it knows that that voxel doesn't have any connections to Wernicke's area, but that one does. And so it allows you to, because it's not making assumptions about the shape or it only bases it on connectivity, it's able to handle this problem. And what that means is pretty significant. So this means that we can parsolate anybody regardless of their shape. And when you combine this with high quality crossing fiber tractography like constrained circle deconvolution, which is what you see here, with edema correction, meaning that we've taken the free water out of the scan before we've processed the DTI, you can actually start to see stuff in the tumor. So this is a low-grade glioma in the SMA, and here are fibers running through that tumor. And here's the language system showing that tumor. Now, in this case, what this is indicating is that there are functional areas and fibers going through this tumor. Now, that doesn't always happen. Sometimes it is just tumor. Sometimes there's tumor on the boundary. And sometimes you get this. What we don't know is that those fibers are doing the same thing. They may be that the body is compensated, but it certainly seems like this is something we're thinking about. One of the questions that we have with a project that we're doing, which Miami's part of, as well as numerous other very world-class centers, is tackling what's called the glioma connectome project, which is aimed to answer this and other questions, which is, what happens if you cut those things out? Does that matter? Does it not matter? We don't know, because we've never been able to see this before, but clearly this raises a pretty significant challenge, which is, well, what do we do now that we know that it appears that the SMA is in the tumor? Now, another question is, how do we actually use this practically? And as Mike pointed out, I wrote a book about this. Part of it was because I couldn't find any books that described really how to use network-based anatomy, and I had to figure a lot of this out on my own because I got very interested in this about 10 years ago. And what we basically thought of, and again, I got this idea talking to Hugh Defoe and kind of modified it because he's not a tractography person, as most people probably know. I personally think that it's always helpful to know, and you can always change your opinion on the reliability of the tractography based on your judgment, but it's never you never really better with less information. But either way, we think about what you're doing in a glioma, you're making a cut between what's going to go and what's going to stay. So why did we pick this cut? Well, the truth of it is this: if you make this cut here, we know that the fibers at the top of the frontal lobe, the anterior frontal lobe, are projecting backwards. So functionally, if you make the cut in the back to remove the back portion of the tumor, you basically have disconnected the frontal pole. That's how it works. And so you might as well get the benefit of oncological resection and supramaximal resection. On the other hand, if you look at the lower left corner, the orbital frontal cortex is not in that diagram, and that's not accidental. It's not an artist's rendering. It's because the orbital frontal cortex connects differently. It connects through the IFOF, and so you actually haven't disconnected that. So what we did there is it's a combination between supramaximal resection, getting the benefit of where we say that, well, if we've made the decision to sacrifice the function, we might as well get the oncologic benefit, but to not make those decisions based on non-anatomical or thinking about the brain as a blob. It's not a blob. We know that that's not true. One of the things about connectomics is because we can look at these networks, we can now see things like the fact in this big frontal glioma that there are no function in this tumor. It's basically just tumor except on that boundary. You look at that medial boundary in the center column, you can see that there's default mode network that's draped over it. Now, the question, of course, now is, can you save it? This is the same as, can you save a facial nerve and a facial nerve schwannoma? Different question. That's a clinical question. But what we know is that this is at risk. And if this patient has some of the symptoms of a DMN dysfunction, which are non-trivial, the DMN is pretty much one of the most important networks of the brain, as I'll mention, then we're not surprised by that. So what this means is not always that you don't take out the tumor. It's that you make those decisions based on data. And so oncofunctional balance means you consider both of them and you decide where to put the line, not that you don't take this out. So in this case, how you would think about this surgery are these three cuts, because you're cutting with three networks. And once you've removed that, then you've basically converted this into a meningioma case, more or less, once you've made those disconnections. But if you think about it in terms of disconnection, number one, you always are getting the adequate resection because you're always up against something that there's a reason you can't cut out. And the second thing about it is you're obviously not going to just accidentally in the middle of this swipe across one of these fiber bundles with the CUSA, because you actually start consciously making that disconnection. And that's a different way to think about it. But this is technically what you're doing, even if you start in the middle of the tumor and work outward. One other thing that we started doing with Connectomics is a novel strategy called interventional neurorehabilitation. And what that term basically means is utilizing things like non-invasive brain stimulation, where we as neurosurgeons actively look at the connectome and utilize non-invasive brain stimulation to try to make patients recover quicker. So for those of you who are not familiar with TMS, it's an FDA-approved treatment. It's approved for depression and OCD. We use it off-label, and it's basically sends a magnetic pulse into the brain. It's painless, and the risk profile of this makes it one of the safest neurological treatments that we know of. What we also know is there's 143 randomized trials covering close to 100,000 patients that basically shows that the safety profile of this, of making people worse, is extraordinarily low. So we began looking at this in an off-label fashion in brain tumors, which the concern had been for a long time that these patients would have seizures because of the brain tumor. And part of this was because this was pioneered by psychiatrists who, would freak out about the fact of having a seizure in their clinic. Whereas for us, we can handle this a little bit more. So we took some risk. We've never had a seizure. And what I can say is that while we don't have class 1 data, I can say that we probably don't need class 1 data for some of the cases we've seen where people have recovered significant amount of function. So the real question is, how do we utilize this to get better results? But I think that it's safe to say that this is very helpful. So how do we pick targets and how do we think through the case? Well, these are complicated. First, you have to look at someone's structural connectome. You have to say what's missing. Because obviously, if you cut the corticospinal tract, there's no way that brain stimulation is going to re-anastomose that. So if you cut the arcuate fasciculus, you might be able to get language to happen around that, but it gets a little bit different of what targets are available, what targets are not available. But also, if you see that there's structurally intact brain, sometimes it's still not communicating correctly. It has abnormal functional connectivity, meaning that the fMRI shows more information on how to tackle this. And when you work together and you look at the connectivity matrix, you look at all their regions and you compare their bold signal, and you look at a correlation. Say, are they firing together? Or are they not firing together? You can start to get a lot of insight into what's called functional connectivity. And the type of data sets that we create with this are huge. You can see that this is a very big graph. What you're looking at is each dot, tiny little dot on here, is a comparison between one of the areas here on the x-axis and one of the areas here on the y-axis. And each one of these are different regions of the brain. So there's hundreds of thousands of comparisons going on. If you look at the time point data, it gets up to about 25 million variables. But the key issue is that by dimensionality reducing this, we can basically look at, and we know who these characters are because we've done a lot of research on it. And so we can start to look at things like saying, what does the language system look like? On the left is a normal language system. It's mostly red. And on the right is an abnormal language system. It's mostly blue because this is a patient who has a tumor inside of Wernicke's area. It's a glioblastoma. So you can actually see that the language system, because it's a network, should be firing together and it should be red, but it's not. And it's not because there's a tumor destroying it. So we can actually see that. But more importantly, each one of these columns is a place in the brain. This is the laryngeomotor cortex, and we can stimulate this and actually try to wake it up. So we created some tools to do this. And what you're looking on the right is a machine learning tool that looks at the functional connectivity data, compares it to normals, hundreds of normals, and tells you what's abnormal. And that's what's in red and blue. Those are areas that are talking too much and too little. So it takes a very complex data set and makes it understandable. It's still not that this is trivial, but what this means is we can start seeing things, like in this case, this is actually a patient with anxiety, but it shows you the example. We can actually start to see the functional connectivity problems in patients and link them to anatomy and take action. Another question is this, getting to the question of how do we preserve cognition? This raises a question of what do we mean by eloquence in the first place? Well, we mean if I cut it, there's a deficit. So how do we look at something like that and start to answer the question? Well, you can basically take the DTI, parcelate it, and make the brain into a graph where each dot is a parcelation and each line is how many tracks go between there. And you can measure things that are measures of what are called Hubness. Hubness is how many different areas are talking to you. So this area has a Hubness score measured by what's called degree 3, because 3 things talk to it. Here, this is a little bit more important because even though it has 3 things talking to it, this guy talks to A, who is a very highly connected guy himself. So this guy is more important than this guy. And this guy, using PageRank, may sound familiar because this is Google, but Google invented this thing in their initial search algorithm. It determines that this is a better PageRank node, more of a hub because it talks to two guys instead of one highly connected guy. And so we started looking at PageRank and saying, who are the parts of the brain that have the highest centrality? Who are the hubs? And I know everyone doesn't know this nomenclature, but I'll walk you through it. Those areas are areas you call eloquent. And these areas are areas that if you really understood it, you probably would think they were eloquent too of what these areas are doing. So these are areas that are very cognitively important. So what this means, and if you do the entire graph, the highest hub in the entire body is the brainstem. So it kind of aligns with what we think eloquence is. Eloquent areas are hubs, meaning that a lot of guys talk to them because they're important, and they're usually because they're connected to the outside world or something like this. But the bottom line is they're critical for signal processing. Furthermore, we know that hubs express different genes, they have different genetics, they use up more energy, and actually they're what Alzheimer's disease targets. So everyone loses fibers as they age, but in Alzheimer's, you lose your hubs. So eloquence means hub. So a paper that we literally got accepted yesterday to Journal of Neuro-Oncology is the question is, what if we have hubs in parts of the brain that most people don't have hubs? Should we not avoid them? And we don't know the answer to that. But we can actually show hubness and show individual variations. So this is actually a group of 87 people. And on the y-axis, this is every area ranked by how much of a hub is it in a normal person. So the brainstem is at the top and some esoteric part of the medial parietal lobe is at the bottom. And then each one of these are different people. So what you're looking at is in the green, in these little lines, we've ranked them and put them as a color. So if you're the number 1 hub of that person, you're green. And if you're the number 379, you're dark red. So what you can see is that almost everyone has the same basic key hubs, brainstem, cerebellum, etc. And then when you get down here, you start seeing that it starts to slowly become redder because there are more, these are not hubs in most people. And it's fairly consistent. I mean, look at this. What's interesting is if you zoom in, which I'll do for you guys, you start seeing these really highly ranked hubs down here in the red in some people, which means that for most people, this is not that important, but for them, that's a hub. If you look at that dark green, this means this is probably ranked in the top 20 hubs of the brain, but it's in a place that you normally don't think about. And so we don't know what the clinical effect of that is. That's what the Glioma Connectome Project, one of its goals is say, does unexpected hubness explain to patients who look fine but don't go back to work because we've actually taken down a system that we didn't understand? Don't know, but it's a way we can measure it and see it. So again, I've cut down my anatomy section for this talk to try to stay under time. I think one of the things about it is, again, if I went through all of my anatomy slides, we could be here for literally 3 to 4 days. I've taught courses that go that long. But in the interest of time, I'm going to try to give you what I think are really the key points of network thinking, because I want to try to give someone something they can take home from this. And people have spent a lot of time recently talking about white matter and bundles and things like this. But the truth of it is, white matter study is a means to an end. I've never had a patient say, "Thank you, doctor, for saving my IFOF," but they have thanked me for saving their speech. And so we care about white matter because it is the backbone of networks and because we care about function. And so saving function is not about saving white matter bundles, and we have to cut white matter to do surgery. It's saving the white matter that hooks up specific brain networks. And Connectomics has the ability to actually show us what fibers are actually going to those networks and which ones are not. And networks are a different way to think about the brain. And what they tend to mean is networks are parts of the brain that are firing on a similar time course. They have functional connectivity. That's why they're red on that graph. They're firing together. And the reason they're doing that is they're usually wired together, and they usually serve a common function. But where function really happens is when these networks work together. So a little part of the default mode network works with a little part of the motor system, and next thing you know, you get something that's much more complex. Now, what's interesting is networks can be subdivided. So depending on how tightly you want these things to be correlated, some things basically correlate with the default mode network, but they correlate more in a little subpart of it. So there's a hierarchy. So the DMN is the top of the hierarchy. But if you subdivide that, the language system falls out of it. We know that language is part of the default mode network. So the simplest way to think through this, without getting extremely detailed into the weeds, is to think about it in what are the main networks of the brain. And there's 7 basic axes. So looking at this diagram, each one of those axes is in a different color. Some of these you're probably very familiar with. So the sensory motor system, including some of the primary auditory cortex, is part of the one network, meaning it mostly fires together, and it fires together more with itself than it does with anybody else. Visual system and the limbic system, including the orbital frontal cortex, which is paralimbic areas, are a network. And there's some networks you probably have spent a lot less time hearing about. So in gold, you have the central executive network. In red, you have the default mode network. In pink, you have the salience network. Believe it or not, this was discovered about 8 years ago, so we didn't know that this existed until very recently, but now we know that it's a really important part of the brain. It's central to depression and a lot of other diseases. And the dorsal attention network. So part of what we wanted to do is say, how do we explain brain anatomy in terms of networks and figure out how these things are wired up and where they exist so that we could show them to people. And the 4 networks that I talked about at the end can best be thought of as control networks. They sit at the top of the brain hierarchy, and they determine how resources get allocated in the brain, what we focus on, what's going on. And this model is a good kind of way to think about how control networks basically work. So The default mode network is your mental state when you're thinking to yourself. The central executive network is an active network. So if you're trying to remember something or focus on a task, this tends to be the part of the brain that's more active. And the salience network switches between them. It's a lot more complicated than this, but this is kind of the basic way to get your thought processes around this. So the default mode network has a central core and then some peripheral parts. The core network is posterior cingulate, anterior cingulate, medial frontal lobe, and a lateral parietal node. The salience network is a cingulo-opercular network. This means the middle cingulate and the anterior insula. And the central executive network kind of looks like this. These are the core parts of the network. There's parts that subdivide across this. So talking about what they do, the default mode network is your internal mental world. It's also fundamental to almost every mental illness, has some degree of problems with this network. The salience network is what brings you out of that kind of living in your own world and awake and active. So things like pain, emotional regulation, etc. And the central executive network is what we think of as executive function. The dorsal attention network helps you keep attention on a task. So if you look at this, you see that we have these 7 networks. And one of the things is that this can look quite random, but there's some logic to it. And so for a long time, we've known that medial frontal injury causes abuli akinesis, but now we kind of have a map of this. So if you look at the SMA, the SMA actually isn't really a motor area, it's a cognitive area. And the majority of it are areas that are part of other networks, part of one of the control networks where they just happen to meet with a motor planning area right in one place. The salience network straddles the middle cingulate and basically connects to the anterior cingulate DMN. And not only have we found this in a study that we published several years ago about anterior butterfly gliomas, where we found that as soon as you touch this in awake surgery, the patients tend to become unresponsive or they tend to become less active. A group at Harvard studied a group of patients with stroke who had akinetic mutism and found that looks pretty close to where the problem is. And so it's destruction of how these two axes work together that probably causes this. And in some patients that requires a bilateral injury, but in some people that causes a unilateral injury. Either way, this is probably something we should start thinking about as quote unquote eloquent brain. It seems to be important. Another thing Connectomics has taught us is that this model that we learned about language is just not enough to explain everything we know about language. One of the things that people have talked about is the concept of a 2-pathway model, where there's a second pathway that is the ventral pathway that handles semantic function, meaning what do words mean and how do you match them to objects, etc. And again, I could give an entire hour talk about this, but I'll show that there's some very interesting additions that we can add and Connectomics have added. One of them was 55B, and I've worked on this with Eddie Chang for a while, and back and forth we've kind of exchanged ideas, and he managed to leverage his into a cell paper. I managed to leverage some of my ideas into a paper, a case report in neurosurgery. So obviously he as usual, Eddie's outshined me in all ways. But the point is, what the data seem to suggest that this is a motor cortex for the larynx, and it doesn't connect to Broca's area directly. And we didn't know it existed. We actually have a case of a patient with apraxia of speech, and this is the only thing that we took out. So it seems to be real, and Eddie has some pretty compelling data on this as well. So if you start looking at all of the things that contribute to making normal speech and normal language, notice that they're different things, we start getting a much more complicated picture. And we just published a review on this in JNS. And basically, this is kind of a closer model of all the things that probably work to make your speech both fluid and fluent and comprehensible and have good syntax, etc. So this is a lot more complicated of a model. And the only way to really display this kind of model is to have the kind of software that can really go into this and find these areas and show them to you. And as we begin to start looking at cognition, what we have to understand is that these things like math and reading and things like this are amazing that they happen. They happen because different networks talk to each other, and there's specific white matter bundles that help parts of the DMN talk to parts of the CN in certain combinations for certain periods of time. So I know that was a rush thing, but I want to leave you with something that it's hard to unsee. So if you look at this, you say, how in God's name could I possibly learn all this? And I'll give you one thing that when you look at this, it looks chaotic. You know, the motor system looks pretty clear and the visual system looks pretty clear, but the control networks are all over here. And how can I memorize this? Well, it turns out that this is not random. So let me show you what's really fascinating. Okay, so if we go through the 7 networks, CEN is in gold, DMN in red, salience in pink, DAN in green. Okay, so if you look on the medial side, you see that pattern. That is, it looks random, but it's pretty striking that looks like something that's a repetitive subunit. Furthermore, when we start looking at the DMN, you notice that you have this pattern where you first go from posterior cingulate to anterior cingulate. You come onto the lateral hemisphere. You go down to the opercular and insular region, over to the parietal lobe, and down to the temporal lobe, like an S. Now, look at the CN. You start around the posterior cingulate, you head anteriorly, you come onto the surface, you go to the parietal lobe, and you come down to the temporal lobe, like an S. And if you go to the salience network, posterior, anterior, insula, parietal, temporal, like an S. Now, the dorsal tension network is a phylogenetically older network, but it has sort of a partial S. Now, it would seem that that's kind of a coincidence until you look at how do these networks express in the cerebellum. And this is work by Randy Buckner at Harvard. And they were trying to find this in the cerebellum, and they mapped these networks to the cerebellum. And what became really fascinating is when they looked at this in the sagittal plane. So if you look at this, if you go around starting on like a clock face, you have blue, that's sensory motor, salience, CEN, DMN, it flips around its mirror image, DMN, CEN, dorsal tension, which in honesty is also on the other side as well in the same rough position, salience, and motor. Again, it's also represented in the flocculus. And when you look at the caudate and putamen, you see that they also have subdivisions of these networks that are often repeating. So what this probably means is that these are subunits that got added in phylogeny at different stages as modules, and these modules became linked to common patterns in the deep structures and are repetitive. And in fact, this has been traced all the way to marmosets. So pretty much throughout at least primate evolution of the brain, the repeated development of these subunits have been put into there, which again is an amazing observation when you see it. So getting out of the scientific back to the operating room, Mike mentioned this book, which I think I put a lot of effort into doing, and the goal is to really think about how do we describe disconnections. Now, it doesn't mean you have to always do a lobectomy. This is a concept of how do you cut, what are safe cuts that you can make with networks. In some cases, these were early stages. It may be that these aren't the right ways to cut the brain, but these are ones that seem to have worked reasonably well for me over the years and make sense anatomically. We have a product that's going through the FDA now. There's going to be a talk at the CNS in the coming weeks where we demo how to use this. But basically the thought is that in the software, what we created was a workflow so that people didn't have to necessarily understand every single piece of the anatomy to get this to work. You basically click a button of what part of the brain you want to be in, and it makes the objects for you, and eventually sets this up to export to image guidance systems. And this is built and integrated with the kind of the concepts in the book. So I said, okay, well, if everyone can't, doesn't really want to try to understand the anatomy like I have, which I understand 100% because I've spent, know how much time I've spent learning this, then how can we leverage that kind of knowledge to make this simple? So I said, okay, these are the things that I would want if I was doing this surgery and doing this cut. We built that into the software. And ultimately, when you look at a tumor like this, you could go into this tumor and start sucking out the center of it and hope you don't hit the ACA, or you can think about it in terms of cuts, where you disconnect the brain from something functional, and eventually, again, removing everything that's disconnected ahead of time. Now, you notice that we didn't remove the basal ganglia. Again, probably safe to say that it's a hub of the brain, and we know that that's generally not a good idea. If you look at temporal lobe surgery, we've known a couple things for a while. The first we know is that you could go about 7 centimeters back on the right, but we know that now why that is the case is that the language system is going posteriorly into the arcuate, and this is why that's safe. But the truth is we know that that rule fails sometimes, and part of it is because people have different anatomy relative to the tumor. The other thing we think about with the disconnection is, how do you know when you're across the cut? How do you know when you're clear? And a really good way to think about the temporal lobe is think about them in terms of three systems. There's the the red system, which is the kind of arcuate SLF system that's lateral. IFOF is, for example, in that system. You have the medial system, which is fornicele hippocampus, and then the inferior system, which is ILF. And essentially, tumors, until they get very advanced in the temporal lobe, tend to remain in one or the other. They don't tend to cross very much. They can, but they tend not to do that. So, for example, this system tends to, these two areas tend to be linked. These two areas less common. And what you know is when you're in the ventricle, you're clear of the system. So what you're basically trying to do with this cut is to parallel the language system until you reach the ventricle, and to figure out where can you cut the STG. And once you've done that, everything in front of this is now basically a meningioma and can be removed. Now, sometimes you can't do this. Sometimes you have to tailor this, et cetera, et cetera, et cetera. We could spend an entire course going through all the nuances of it. The temporal disconnection is a concept saying that if you had to remove the temporal lobe and everything, this is what you can do. And this could be part of supramaximal resection or not. The one thing I will say, though, is that the temporal pole is, again, a non-trivial part of the brain. If you go back to the hubs, this area here in the left temporal lobe, left TGV, is one of the hubs of the brain. It's up in the top 15. And we know that it's critical for integrating areas, and we know that semantic dementia as part of frontotemporal dementia results from atrophy of that part of the brain. So again, it has a complex anatomy, but of course, if it's been destroyed by tumor, or you're going to cut it out in the process of getting the tumor, then you may make the decision to say, this is just life, I'm sorry. But it gives us an idea to think, what are we doing wrong? How do we do, if we're doing epilepsy cases, do we need to remove that part of the brain? And we might do something different than an ATL and change our anatomy to accommodate certain connections. So if you look at a temporal lobe case, how do you incorporate that into this nasty insular glioma? You can see this is insula, this is the temporal lobe. And here's the anatomy that we have. Now, again, here in the red and green is the neglect system. We now know the anatomy of that. This is what it looks like. You can see that in this case that the IFOF is attenuated and it's really distorted by the tumor. This is the normal IFOF. And again, you can see CSD trichrography provides really good connections because it can handle crossing fibers in a way that DTI cannot. If you look over here, you can see that here's the corticospinal tract running over the top of the tumor. So while you could hit that, it at least gives you an idea of what you can take, what you can't. So you know that, for example, this is face motor cortex, and you need to make these cuts. Another angle is the medial parietal, and I'll finish with this. So for example, if you're looking at this tumor, this is very complicated. You may not be able to remove this safely, but at least now we can see the anatomy. And you can see that, for example, the default node network this thing is wrapped around it. And this is probably one of the main reasons these patients have so many cognitive problems. But if you were to attempt to tackle this, and again, this is a clinical decision, not a radiographic decision. These are the cuts, and you can see that this is a numerous number of cuts, but what you're basically trying to do is to disconnect this from the networks as best you can. And I'll kind of skip the video in the interest of time. Anyway, thanks everyone. I know that was a lot. I was trying to give you as much information as I could in 45 minutes.
Awesome. Thanks so much, Mike. That's really phenomenal work that you're doing there. Let me just answer a couple of questions here before we get to the cases. Manuel talks about when you're looking at the white matter and the connections, how do you know if when you're taking away some of the tracks, if you're just decreasing the neuron transmission efficiency, or if that matters versus cutting the whole track? So if you cut partial tracks, I think what he's trying to say, how do you know is if you cut a partial track, when is it enough too much for complete cutting of the track? Can you predict?
Well, of course, that's the $1 million question. But I think one thing we have to think about is, let's say we have a track bundle. That's your track bundle. It isn't the same as a peripheral nerve. So if you look at the median nerve and you say, okay, if I cut half of it, I'm cutting half of the motor fibers down. That's not really how it works. The best way to think about it is a series of bus routes going through a city. So they're going to spread out And then they're going to kind of come in when they go through a central tunnel, and then they spread back out. So maybe like a better analogy would be, one of the main subways going through the center of Manhattan. It fans out at one part in the Bronx, it fans out another part in Brooklyn, but when they run together, they're on a common set of lines because there's only so many ways to go through there. So if you were to cut half of the IFOF, it isn't that you've reduced everything that's going through it by half. You removed all of one part of it and none of another part. So one of the things that we have to think about is that bundle is actually just running together for convenience. It isn't a homogeneous structure. I think to get into the question of specifically how do we practically do this, one of the things you have to think about is we don't have a real great metric for putting numbers on this right now. So how do we know how good of a job we did? And of course, getting into the question he has, which is if someone has a deficit, is it a functional disturbance or a structural disturbance or both? You have to have a way to quantify that because otherwise we can't iterate. So what we started working on is a machine learning metric called the NIS, the Neurological Impact Score, or the Network Impact Score. And what the Network Impact Score says is it puts a number not on the track, but on the default mode network or the central executive network. So how much of that network have you destroyed? And remember that you could destroy a network by moving the cortex or the tracks or both. And how do you have a metric that accounts for the fact that the track could be there, but it's not really, it's injured? That's a very complicated question, but it's a question for data and machine learning, and we should be rolling that out over the next few months.
Yeah, that was actually one of my questions is when you're looking at some of these tracks, I mean, obviously they're duplicated in the brain in the left and right side. And we know obviously that they have different functions on both sides because of just what we know about the brain. And so, and I think I'll save some of the questions for my case, but when we talk about some of these left mesial structure, like left mesial temporal lobe, right mesial temporal lobe, what is the importance of memory in both sides? Is it weighted to one side or the other? Do you think you could use the NIST score that you're developing to kind of help decide, how these left and right areas are altered and maybe compensating for a tumor or epilepsy that may be happening in a destructed area?
Well, and that's one potential answer. I think the NIST's clear goal is to say, how do we measure our results? And how do we know that this is destroyed and don't bother trying to save it versus go for it? And of course, that which is not measured is not improved. Let me actually show you a second thing, and it's not part of my talk, but I think it's pretty cool. So pull this up here. So one potential answer to what you're talking about are what are called graphs. So I touched on this with PageRank centrality, but let me share my screen here. I want to do that. Okay. So think about the brain as a graph, and if you put an insult in that graph, what you're hoping is that some way, somehow, the information can wind its way through the rest of the path. So if you put a hole here, it can go around that. And probably with language, that happens sometimes. So there's probably some times that we destroy part of the language system, or a tumor destroys it, and then over time, over a year, the patient upregulates the connection, strengthens through synaptic plasticity. What we don't have is a real way to dig this. And how do we know that ahead of time? So what we started doing work on is a question called path length. So if you were to say, I need to get a signal from A to B, and all of these things are different parts of the brain, and these numbers are how many fibers go between them. Well, the shortest path is something like that. And what you just said is, okay, this is the one that has the most connections to get to A to B. But what if we do that? Well, it's going to change the path length because you have to go on a different route. And this may be just too slow to get the job done. Now, why does path length matter? Well, it turns out that there's a lot of data that links path length in the brain to IQ. So it's reasonable to think that if you lengthen the path of the surgery, you're probably making somebody less intelligent, or at least less able to handle fluid calculations. But we don't know where that threshold is. But what we started looking at is we set up an algorithm, and we've been messing around with it for over a year now, where we can basically do theoretical brain surgery. So we take the DTI, parcelate it, make a graph, and then we basically calculate path length for the whole brain. We can calculate a number called global efficiency, which is the measure of all paths in the brain. So if you're really efficient, you have a lot of short paths. And we know that on average, you tend to be smarter. So Well, then what we did is we said, okay, let's make an algorithm that does every possible brain surgery, not everything that's possible to do. So we don't, remove one parsolation here and one parsolation here, and that's not going to happen. We set it up where it could only do surgeries of continuous areas on one lobe. And then we did the same thing to the graph, and then we calculated efficiency and see how it changed. This is where it gets super interesting. So what we found is that different people have different tolerances for it. And so this is what we call a stepwise lobe. And so a stepwise lobe is this is what's normal. This is what happens if you remove one area, 2 areas, 3 areas. What you notice is a smooth curve, meaning that there's no way that 4 is ever better than 2. This is a different individual in the same lobe. And you notice that there are some situations that a 4 is better than a 2, meaning that this is more bad than anything here, or some of the things here. And that is a big deal because this means we want to know who this is, because that's actually much more damaging than. So this person only matters how much you take out, and this matters what you take out. We've now done this on several hundred brains. This holds. It holds if you do bilateral, unilateral, however you do it. And whether this predicts compensation, again, is a scientific clinical question that we just don't have the data for right now. This is one of the things we would look at. But whether this tracks with clinical outcomes. So I think it highlights a key point, which is tolerability. It really comes down to, is there a way for the information to navigate the graph of the brain and get from target A to target B if you remove that area? And that's not something that we're going to do with the naked eye. It's computers that have to get involved. Really rational way forward, which we might be able to know that ahead of time. That, wait a minute, hold on, you can't take this, or it's going to just knock everything off. Whereas other people, don't worry about it. Just take out as much as you think you can, and they'll up to here will be fine. And when we start talking about supramaxillary sections, from an oncology standpoint, it makes perfect sense to take margin. We know that gliomas, as much as they tend to be quote unquote diffuse, most of the time at diagnosis, they're mostly close to the tumor. It's just a margin issue, like breast cancer. We just can't take enough of a margin. But of course, the margin in this situation, we're starting to run into networks and other consequences that we may not necessarily understand. And this gives a potential way that we can tackle that.
Awesome. Before we get to any more questions, let's get to some of our cases. Simon, do you want to go?
Yeah, sounds great. You guys seeing that?
Yep.
Yeah, looks good.
Good. So thanks, Mike, for having me. I just want to throw out there too that I've been here and there reading through Michael's book, the glioma book. I'll add my own shameless plug. It's a remarkable accomplishment. I mean, I think the way of looking at the networks, and it's just a whole different kind of view of the brain. And I think it's just incredibly forward thinking, very avant-garde, and it's a real game changer. So just want to thank you, Mike, personally, even though I don't know.
Thanks. Appreciate it.
Fantastic. So I have 2 quick cases, some 2 low-grade glioma cases, and I don't think nearly as cerebral as the stuff that we do, but I would love your insight in the panel, kind of into these cases and what you may have done differently, or just how to interpret some of this data. First case, and real quick, is a 25-year-old female, and she presented with intermittent right face and arm twitching for about a year. So this prompted an MRI, and this is the flare, and you can see this area of abnormality here very clearly delineated. No evidence of enhancement on the post-contrast studies. And I got an fMRI in this case. I could see just anatomically it looked like a virus in front of the hand motor cortex there. So I didn't think there was really anything special. But I found the functional MRI in this case to be interesting, and it changed my approach in this case. And I'll share a video of the fMRI. And I know it's a little bit kind of difficult to see. And this is us scrolling up, and I can kind of pause and get to it. So you can see the hand motor, very robust here, obviously parallel here, I should say. And then corresponding to some of the naming tasks that the patient was given, I thought there was very robust activation of expressive speech, flanking. And, perhaps this betrays a little bit of my own lack of understanding of the connectome, but I was surprised to find such robust speech in that area on the fMRI. And I had planned actually to do this asleep because I'm confident in the level of motor mapping and monitoring that I can do, even though I'm sure many would do this awake. She was a young girl who was anxious, so that kind of pushed me. But when I saw the speech represented this way, I then converted to an awake craniotomy in advance, obviously. And when we stimulated these areas, she had very obvious reproducible speech arrest. And I don't think it really changed much of what I did. This ended up just being kind of an essential spirectomy. It went fine. I could show you the post MRI here, and the image that we had, just, a very classic kind of subpeel thing down to the base, and it went fine. And her speech was preserved. But just kind of wanted some insights into how you would regard those areas. Would we say that they're kind of in this non-canonical category of expressive language area? Would you regard them as supplementary motor area representations of speech, laryngeal motor cortex, even though we're a gyrus in front? And I just kind of wanted to get your insights on that. And also, just in general, I think you would, the panel would agree that this went well, but there are times where delineating the depth of some of these low grades can be very difficult. At that point, the navigation's out the window. And is there really anything that's what I find in my low grade resections could be a challenge, that kind of blend. And when you get into that white matter, it can be very difficult to find, the limit of it. So I just was, looking for some insights there.
Well, so that's a fantastic case. So what you can counter there is 55B. That's exactly where it's located. It's in the precentral sulcus, and it's right in front of the hand motor cortex. So it's in front of it's not in the motor cortex, it's in front of it. And because it sits in the bank of the sulcus, we haven't seen it. But if you look at it, if you go into the fMRI literature, it's always been there. We just didn't take it seriously. We thought it was noise. And so if you read the task-based fMRI literature of speech, it's always lit up. It's always been there. And until the Connectome project, where they found that it is always part of the language task in all of the language tasks. And then we didn't believe it. And then Eddie basically put electrodes on it and showed that it is with a scope down somebody to prove that it's actually the motor cortex and larynx. That's a pretty compelling combination. In your case, the reason she did well is that you preserved that sulcal bank. And we haven't thought about the sulci part of mapping, but the areas are different. The other thing the Connectome project found about 55B is that it's the most variable parsolation. So sometimes it's actually split in half in two parts by the frontal eye field. And so they found that was the hardest one to be certain that they were correct on. But the truth of it is, when we went back and looked at all sorts of ways we do fMRI processing, it's usually there. And so in your case, what you have is, you have a very good evidence of that. And in that situation, it shows you that this is a language case that we would have not normally thought as a language case.
Right.
I'll share you a case that we had really quick. So I did this case 8 years ago, or 6 or 7 years ago, very long time ago. And you can see the tumor, same kind of basic place, not very big. We basically did this asleep, thought this couldn't be anything. And this guy basically this is a grade 3, but he's been under control for years now. Never recurred, never had any complications of radiotherapy. lives alone, takes care of all his ADLs, works as an accountant where he sends advice by email detailing financial transactions, but cannot talk. He can't phonate. And Eddie didn't believe me when I said this, so he sent out a linguist to Oklahoma, and they spent 6 hours with them and said, nope, it's only the larynx. So we wrote this up. It was in neurosurgery a few months ago, and it really shows that we didn't know that was there, but it's there all right. And so that's a really fantastic demonstration that that this is real. It's really a part of the brain.
In this case, is there any phonate? I mean, is he able to make any kind of sound, or it's just complete?
I have a video, and he basically talked. But they did language, tongue, mouth, everything. Everything works except his larynx. So really correct, but everything what he says actually is correct, and is so his language function is normal. This area has its own set of connections to the parietal area and to Wernicke's. It has its own connections, so it's not part of and it doesn't talk to Broca's area at all. We've never found any connections directly hooking them up. So what happens is if you go deep to that and you start messing around here, you can just separate it out. And we didn't know that. We just walked right into this. But we didn't know what happened to him for years. I just thought, this is so strange. Like, what did I do wrong? This is in the language area, blah, blah. Truth of it is, we didn't know better. I didn't know that barrier was there, but it's there, all right. And you kind of proved it. So yeah, that's awesome. I mean, that's a really great case.
Yeah, it was a wild one. Let me go quickly to the other one, because I know the other guys want to present. Hold on, let me reshare. Okay, is that up there, case 2?
Yep.
Great. So this is a 51-year-old male, very sudden onset paresthesias in the right face and arm. Resolved, and then 1 day later came back. Otherwise, before this, had no issues, totally normal guy, works as a construction worker, but had no complaints. So came in, got this MRI. I'm obviously just showing you a slice here, so a large left insular low grade here with significant temporal involvement as well. So here's there actually were 2 punctate areas of enhancement as well. Here's the FMR. So yeah, let me just back up a little bit. So what stood out to me in the radiologist here was what we interpreted as arcuate or SLF, and how much it seemed to kind of run into this tumor. Not obviously all the way anterior, but the anterior extent here is pretty dramatic. And, ultimately I'll show you the sagittal here, where the radiologist ended up kind of marking some of this as well. Very kind of thick and robust, and obviously here's a tumor, but it really kind of projected into the mass. I did this operation with the patient awake. He's bilingual, Portuguese and English speaking. Basically, every single area that I stimulated along the operculum led to speech arrest. It was a remarkable number of positive gyri, except that the superior temporal gyrus was pretty silent. So I was able to take that and kind of get into it. But, one of your illustrations from the book and what you had earlier in the talk, where you're just kind of retracting up on the operculum to get into the insula, is how I did this. It became very challenging to see far forward and up to do it. And he did have pretty significant issues after the operation with language. Here are just some basic questions pertaining to this case and that fMRI. I mean, obviously the first question here, you have your own system and with Omniscient and just your experience with looking at DTI. But, I think for those of us out there who, do DTI with some degree of frequency, I think there's still a challenge in pinning down kind of what these fiber tracks are, even for the neuroradiologists who are performing these studies. And, if you were to encounter something like this, with that level of overlap, if you will, how much is that influencing you beforehand in terms of what you think you can remove, or how closely are you paying attention to that during the actual operation? And, I mentioned here too, since there appear to be fibers connecting with the SLF posterior to the main area of tumor overlap, does that area of presumed intact connectome embolden you to get a little bit more aggressive, thinking that, you know, there may be some redundancy? And probably the idea of redundancy is probably moot based on your, you know, connectomic impression of the brain, but you know, it's still a little bit rudimentary, but that's, you know, a basic concept that I've kind of adduced in these circumstances. And just this general idea that I think we've kind of, you know, grown somewhat accustomed to the concept of low-grade gliomas displacing fiber tracts and not so much invading. Now, obviously, you just showed a few examples in GBM and the splenium there, where that's just not the case at all. But you know, just in general, you don't have to necessarily answer the question so specifically, but a sense of kind of what this case, how you might have handled this case, and some of your thoughts on what these fibers represent and how you may have, you know, respected them or violated them accordingly.
Well, yeah, I mean, the answer is this. This is the kind of stuff that we need to, as neurosurgeons, to be talking about more often, which is not when we had some case that we got a GTR, it looks phenomenal, but the case where you had to go and get a brutal stalemate to get 60% out. Because that's the cases where you really, really understand where the challenges are. I think one of the first things is this: the arcuate you have looks accurate, and one of the challenges you have to understand, that's the difference between a tract and a network, where you don't know which fiber is actually, because there's other things running in the arcuate, right? The arcuate connects parts of the operculum to other parts of the operculum that have nothing to do with language. Mm-hmm. What the tractography in this situation helps you to say is, hey, there's probably a problem here at getting some of this operculum off, and it's going to be somewhere around here where I'm likely to encounter this. So I might have to have a backup plan to not go transopercular. It's obviously easier to remove the operculum. It's very hard to get up to the opercular sulcus in insular gliomas with the operculum on there. I mean, it's possible, but it's a lot easier when you have some of it off. But in this case, you just say, man, that's bad. I might not be able to get that out. And remember that the information has to get there somehow. So what we found in a paper we published in Operative Neurosurgery several years ago is the DTI isn't millimetrically accurate, but you should ignore it at your own peril. It's pretty, it's going to be around there somewhere. What DTI does in the situation is it frames your thinking. So if you think about the brain as this amorphous blob, the challenge in glioma surgery is constantly putting anatomy into this situation because you don't see it and it's extremely complicated. So what it allows you to do is to put anatomy into your thinking so you can think about this like you do any other surgery. And it's a constant struggle because, you know, we know how complicated the cerebrum really is. As far as how often are we seeing fiber tracts in the low grade? And the challenge with it always was, how do you know that if there's no tracts, that there's nothing in there? And without free water correction, it's challenging because sometimes you just see a hole there and you don't know whether this is or there or not. What I will say is that when we've seen cases where we've had free water correction, and we haven't had it that long, there are some cases where the IFOF is smoked and the patient seems to be doing fine without it. And it's usually those tend to be oligos. Things like astros or some of these other things often can have stuff running through them. And there can be a mixture of some pure tumor and some infiltrated part. So the answer is, I don't have a knowledge, exact number because we just haven't had it long enough to have enough numbers, but my gut is somewhere like 60-30, and it really depends on how dense the tumor is. If it's just pure gray mush, it's probably not functional, but there's certainly stuff on the boundary that's just going to be basically brain with some tumor in it.
Right. I mean, it was a grade 3 anaplastic astro, and a lot of it. I'll tell you that there was really, in this case, I mean, I'm sure you've encountered this too. I was really able to develop quite a plane around a lot of it. I mean, it was just very difficult at those kind of superior and anterior reaches with just retracting the operculum. But, there were definitely significant functional, really language-related issues after the operation. We had a neuropsychologist evaluating him before and after, and he's recovered, but, definitely took a hit.
It always takes balls to do language cases because they're going to look bad, if nothing else, because of swelling. But with rehab and even with TMS too now, you can often get these patients significantly better, but it never feels good when you have one of these, brutal stalemate cases. But you just have to trust that if you did it right, they will usually get better over time.
Yeah. Thanks again.
Thanks, Simon. Great cases. And great outcomes as usual. So while Dan gets his cases up, two quick questions is one in that last case that Simon presented, when you're talking about the SLF along the back side of it, there's a couple questions coming in do you just go based off of the DTI, or are you doing additional mapping and throughout these cases for some of these non-motor, non-speech pathways? Or are you totally relying on the imaging to kind of stay out of them if you feel like they're important?
Both. You want to use both. Again, if Mitch Berger taught me one thing, is that if you have two adjuncts, they can both improve the patient's care. What's the benefit of using one? And so, yeah, there's times you can bring in technology that doesn't add anything to it. But remember that network anatomy doesn't always mean essentiality. And we're still working on trying to build up a concept of essentiality inside of connectomics, but we're just not there yet. In the meantime, the why, then the question is, if mapping tells you essentiality, why not just do mapping? And the challenge is, I just think that maybe there's people who are brilliant minds and smarter than I am on this, but I personally don't find that when they're in the middle of the deep white matter, that sticking a probe in there gives me knowledge that I really can absolutely say this is certain. And having the image guidance will have brain shift, but the brain shift in the white matter is not that much. It doesn't move as much as you think it does. And at least you have some idea, mental framework of what I think is basically going on and where I should use stimulation. So combining those two, and what you just have to remember is neurosurgery is a really hard job, and we should be humble and understand that we need every piece of advantage we can get. We're not all perfect here. I mean, this is a difficult problem. So I use everything I possibly can.
Sure. Okay, Dan?
Okay, can you hear me and see my screen?
Yeah, looks good.
We can indeed.
Perfect. All right, well, Mike, thank you so much for an amazing talk, as well as hosting me for 2 weeks when I came down to Australia and took this picture. I really think it takes someone very special not only to understand and pioneer this very complicated area, but also to simplify it, both through textbooks and through the Omniscient software to enable sort of the rest of us to understand this very important and burgeoning field. So thank you for your efforts. So I have a case to present. This is a 52-year-old male. About a year ago, he had a resection of a left frontal anaplastic astrocytoma at an outside hospital. He also had 36 sessions of radiation therapy and chemotherapy. And he presented to us neuro intact, just with a concerning surveillance MRI finding showing increased flare volume near the resection cavity. So you can see here in the bottom left corner, just sort of increasing flare volume in sort of the left superior frontal gyrus. Really no contrast enhancement, but just over serial scans, increased flare volume. So intraop, we did the surgery awake. Motor mapping was negative for any movement arrest or generation. We resected the superior and middle frontal gyri on the left side. Subcortical mapping was negative, and the patient's speech during surgery showed some hesitation, but didn't really change substantially during stimulation. And then postoperatively, he awoke completely aphasic. The aphasia slowly improved, and at discharge and at 2-week follow-up, the aphasia was almost to baseline, although not yet completely to baseline. This is his, postoperative scan. We resected the entire flare-enhancing area. And, one of the important questions that we always have whenever patients wake up aphasic or hemiplegic is, is this patient going to recover? Is this, in our sort of probably oversimplified algorithm, is this SMA syndrome, which should get better, or is this a permanent neurological deficit? To us, meaning that the motor cortex is permanently injured. And we've been playing around with our Omniscient technology and trying to see if the Omniscient can help us predict recovery or not. And I borrowed the slide from you. We think about the initiation access, which includes the SMA, the salience network, and the default mode network. Injury to this area prevents people from initiating goals into plans. So was, maybe in our minds, if there's an injury to this initiation access, that could potentially recover, versus if it's motor cortex, maybe, or speech cortex, or speech network, it wouldn't necessarily recover. So the Omniscient software tells us what parcelations are missing, and this person's missing parcelations were frontal language, sort of the left inferior frontal gyrus, parcelations 45, 47L, and IFJ. all of the parcelations were present in the default mode network, the salience network, and the SMA. So this potentially would lead us to maybe believe that this patient would not recover, although in fact they did recover. And then we sort of looked at both pre-op and post-op networks. So on the left is the default mode network. Looks relatively, even though all the parcelations are present, it looks, the parcellation voxels look smaller. There's maybe fewer white matter connections. Same thing with the salience network, just sort of looks less robust. There's fewer connections. SMA, same thing. The parcellations are present here for SMA, but there's just sort of fewer and less robust connections between both sides. And then you look at the language system, and I guess this was a little bit surprising. So sort of what looks like below the level of the resection, 45, 47L and IFJA are missing. Maybe they were sort of undercut and disconnected. So the lobe is physically present, but they're just sort of disconnected. I'm not totally sure. But language looks relatively preserved around the area of the resection. So I guess the main question I have is, do you think this sort of seems like a picture of initiation access injury, which can potentially get better? Do you think that maybe we need more machine learning to understand this, but the fact that the speech areas 45, 47L, IHAA are missing, does that mean that you may be less likely to fully recover? Do you think that, so you wrote a paper about SMA syndrome improving if you preserve the crossed FAT fibers. Does that concept exist for the other areas of the initiation axis as well as the salience and the default mode network? Is there sort of contralateral compensation if you preserve the crossing fibers? And then finally, do you think that SMA syndrome is sort of an incomplete or misnomer, and should we be calling it the initiation axis syndrome rather than SMA syndrome?
I think, yeah, I mean, there's a lot of questions there, but I think that the key issue is part of the main, again, we've known for a long time SMA syndrome tends to get better, but that number is not 100% or 99%. There's about 10% of people who never really improve. And that's even if you have a binary like improve yay or improve nay, which is not really very sophisticated. What we know is that a lot of people have some, it's not trivial to remove the SMA. That's an initiation site. We know that at minimum, there has to be something else that takes over. And our best theory is those are the crossing fibers of the crossed FAT. And if you go back to your image, you definitely seem to preserve some of those crossed fibers. And you also seem to preserve if you look at, see that, for example, there's what probably happened is you trim back some of the SMA and then he's learning slowly to utilize the other side. As far as the other, the missing parcellations, it's something we can help you guys with. It's a technical issue about the voxel size, and we can correct some of that. We were talking about this the other day, but what it really comes down to is, in this particular case, is what is the minimum necessary to get a recovery? We don't know the answer to that yet. That's a glioma connectome thing. I think that one of the real ways that we can start to learn this, though, is start to have numbers on it. How much of the SMA did you actually take out? How much of the initiation axis did you take out? If you look at this case, you still preserve the anatomic framework of the default mode, but it could be, working or not, right? So then there's some fibers there, the parcellations are still there, and that's great. And that may be enough for that to function. The salience network in this particular case is relatively posterior, and it's still connected. And that's an important part of initiation as well. So what we don't know is exactly what do you have to do. What we do know is we're starting to get an idea that, for example, if you cut the corpus callosum in the process of taking out the SMA, it's hard to understand how that would get better. And in my experience, the only two cases that I've had in my collection where they didn't get better, that's what we did. We didn't know better, and and didn't think out exactly how would the SMA improve. You don't want to cause an SMA. It's not a trivial injury. It slows down the introduction of chemotherapy and radiation in addition to quality of life. But if you're going to cause it, you need to have a path that will get better. And so my best model for this is, at the minimum, one of the SMAs needs to be hooked to some of the motor planning areas of the lateral frontal lobe. And if you do that, it seems to be that you're not going to get SMA syndrome, and if you do, it's going to be very, very mild. We have a paper that's being reviewed at, I forget, one of the journals. It seems like it's been in there forever, showing that, you know, when we became aware of the FAT and started having on our image guidance, we stopped having SMA syndrome for the most part. So it's important to think, if you're going to cause it, at least make sure it gets better. And the next question is what is the relative benefit of the additional 3% resection that you get on that back end? Is that worth it? You know, because you start getting back there, there's something that has to fire. If the motor cortex is floating in space and there's nothing driving it, it won't work. So there has to be something there. And it doesn't mean that in certain cases, taking out and causing an SMA syndrome may be life-saving. But if you're talking about a high-grade glioma, that may be that they don't get into chemo radiation quick enough, depending on the context. So we always have to work this into a continuum of neurooncologic care. But yeah, I mean, that's a very, very interesting case. But I think you guys skirted the tree line and got away with it. So that's, you know,
yeah, no, I think that thanks for presenting that case, Dan. That was a good case. And, you know, I think the other discussion of this case was when we watched the tumor growing preoperatively, you know, it was growing at a very slow rate, and patient was asymptomatic, and the question of whether to just change treatment versus a re-resection was brought up. And because it was so close to the SMA and to these areas, we knew that if it continued to grow, we wouldn't have been able to kind of get away with this kind of surgery. And that therefore pushed us more towards a surgical approach rather than a just changing treatment and seeing how he would do treatment. And so,
I mean,
I remember your guidelines as not to take the corpus callosum with the SMA with the fat. So we were very careful.
You guys did a fantastic job saving the cingulate gyrus as well. That's pretty solid. Yeah. And that's, you know, that's probably why he's recovering. You've given him enough framework to initiate at some point. And I don't think any reasonable person would say, hey, I don't want to have a deficit for a couple of weeks to survive. But what you don't want to do is you basically get a great resection, but then they wish they were dead because they can't do anything. And we haven't really spent a lot of time thinking about the medial frontal lobe, but I can tell you just on my gut experience, this is the place where I've had these cases where they wouldn't count on the score of hemiplegia, but you don't feel good about yourself.
Right. For sure. All right. Let me share my screen there too for the last quick. I'll try to make mine quick. Mine's a little bit more technically less challenging than the last two. So, but thanks again, Mike, for talking to us. It's just an amazing amount of work that I think most people would take a lifetime to do, and you've done it in such a short time that you should really be congratulated. This is a case, you know, I'm not only am I doing the tumors, I'm doing some epilepsy work here at the University of Miami, and this is a case that kind of combines both of them, and it really highlights the importance, I think, of understanding the connectome, because in this case we had a really young patient, 22-year-old, with a very low-grade tumor and epilepsy that was refractory to medicine, and I think these are the cases where the patient's going to live for another 60, 70 years, and any kind of subtle deficits is really going to be amplified. This is early on in my practice, and I think now I probably would have done this case a little bit differently, but this is the scan. So he presented with epilepsy that was localizing to both the right temporal, anterior, and posterior lobe, and had this small flare abnormality in the posterior mesial kind of temporal lobe behind hippocampus. Here, very minimally enhancement, and it had already been watched for like 2 or 3 years, really not changing, but he'd already been maxed out on 3 epilepsy agents and was still having seizures 2 to 3 times a week. And so the recommendation was then to do surgery. And so here's the coronal scan here, just showing kind of how tucked in underneath it would be in, I guess, the blue area of your talk, Mike. We got a functional MRI just to confirm dominance, which was obviously on the left side in this gentleman. This was before we had DTI tractography, but we also do here is we do PET scan for our epilepsy cases, which showed that there was perhaps some interdictal focus in the lesion. We also looked at the volumetric analysis of the hippocampus, and we found that the right side was slightly enlarged, but not within significant value. And then, you know, for epilepsy cases, they spend a lot of time for neuropsychological evaluation. And I'm not sure why that hasn't taken off for tumor surgery as much as we would like. But So we looked at his emotional behavior. We looked at his intelligence. He completed high school. He had great intelligence. We looked at the dominance on the right and left side, and we say that basically he has good verbal memory scores on both sides and intellect on both sides. And the red is the blue is the right side, and the gray is the left side for both visual and verbal memory scores. So I'm not going to go through all the details, but just both sides were working well in this gentleman. And so so the idea was that basically, he has seizures. We know it's coming from the right side. EEG was localizing to it, and he has a fairly good cognitive score. And so the question is what do you do here? What is your surgical approach? Is it tackle the lesion, or do a right anterior temporal lobectomy, do laser to just the lesion for the actual area itself? How do you know exactly what to tell this guy who's just about to start working in the workforce? What is going to be the impact of his visual and auditory learning and memory? And so I think how would you tackle this now? I guess with the Connectome and the Omniscient kind of software with making some of these decisions.
Well, I think the first step is that we have to acknowledge that the neuroscientific literature has shown that the temporal pole is not some just, piece of temporalis muscle. It's doing something important. Now, that doesn't mean you can't get away with it. We know we can, but there's not that there's no consequences for taking it out, because it's in a major integrative site. It's a major hub of the brain. So I think that given the experience with LIT, and it just comes down to the discussion, but the truth is, if you swing and miss with LIT, it's not, you end up doing the open operation you do anyway, and you haven't lost a whole lot. So I would personally try LIT first and see what happens. The question then, of course, is do you do the hippocampus or the amygdala, or both, and or just the lesion? And of course, that's the $1 million question. I think that one of the challenges with it is we're still digging into this, and I have a student this is their project this year, which is talk about getting into the hemispheric asymmetry issue with connectomics. People have done it, but what specifically is different? And it turns out there's not much. There's only a handful of parsolations that change affiliation between the sides, and none of them are in the temporal lobe. And so it's interesting when you say, how would you assess the load of this? Well, one possible explanation is graph theory, and where people have done some studies showing that if you were to take the lesion, or let's say where the focus is, and you were to go and map out a graph of everything that's connected to, and say, I'm only going to take out things that have this much connectivity to that area in that graph, whatever that is, you actually get better seizure results, at least on a very small level. Now, this is with the ECOG and not fMRI, and so make sure we understand that. But the second thought process is that at least would tell you what you need to disconnect, because again, ultimately, you don't necessarily need to destroy the entire hippocampus. There's probably just a couple fibers that need to get cut to take this thing offline, so to speak. And so this may be one of the situations where we start to say, okay, how do we dig into this question, utilize machine learning to say, given that lesion, given the EEG data, and given what we know about what happened to the seizure patient after all of this, can machine learning find what do you need to do exactly? What gets the results? Where is the actual epilepsy zone? Not the onset zone, but the whole zone that needs to come out. And of course, then the other question is can they handle the load on the other side? These are questions that I think that are going to be very, very challenging to answer without really taking a machine learning approach. Machines can learn patterns, but we need data, and we need to have, where was the lesion? What does the connectome look like? And then let a machine sit down and figure out what we need to do. But I think that there's some critical questions in this. As for how I would handle this in 2020 without having that kind of information, again, I would try LIT and then see what happened. And then if necessary, then you can discuss ATL and this other thing. But I think that in general, and maybe people can update me on this, but the last time I really engaged into this literature, it seemed to me that the cognitive side effects of LIT for the hippocampus were less than if you take out the whole lobe, and less than if you even did a selective, which does involve manipulating and cutting some of those connections. You got to get in there somehow if you're doing surgery. And I think that in general, we should be a little bit thoughtful, saying, well, some of those cognitive problems may be from the hippocampus and some may be from the ATL, the anterior temporal lobe.
Yeah, no, and I agree. And I think that if this came across my clinic today, that's probably what I would have really pushed for. We had that discussion, I think this was like 6 years ago or 5 years ago. We had the discussion then about lit versus ATL versus just legionectomy. And based on all of the EEG data that they had at the time, they were not convinced that there was not some really anterior temporal pole involvement. And that's what kind of skewed them, that it wasn't just the mesial part, it was not. And then it's a question of this, do you take the advice of trying to just do the most minimally invasive thing with in our eyes, it makes sense just to do something more minimally invasive, less risky up front, with the idea of you could always come back and fix it. But of course, not all patients want to go through 2 brain surgeries, and some do and some don't. So this gentleman ended up getting an ATL with a full resection. Luckily he did well. It was ganglioma. I saw him back recently. He's seizure free for 5 years. Tumor's obviously not coming back. He is working. He still has his baseline ADHD, and still has some struggles with cognitive kind of abilities, but not really much different than preoperatively. Would that be different if we were more minimally invasive? It's hard to say. It's hard to say, but
Yeah,
I think that's where we need to look at this and understand those.
And there's a couple points on it, which is, of course, we should be measuring neuropsych testing. One of the challenges is all the gliomas, most of them can't make it in there. And the second is, even the ones who can, like some of them just can't sit through a neuropsych battery. One of the things that we put on the Omniscient platform, the reason for the glioma connectome project, but it could scale to anything, which is the descale the neuropsych testing to where it can be done by a non-expert and it can be processed and scored in the cloud to leverage the expertise of machine learning on this. And I actually got that idea from Sean Grady, and he's like, I can't get it very easily here at UPenn because there's just not enough neuropsychologists to test all the people we want tested. But getting some data is probably better than getting no data. And if we sit there and hold out for only the best possible neuropsych test, we're not getting any information on the patients. So I think one of the things is if you sit down and really talk to people, there's a lot of people who they're going about their life, they're okay. But if you talk to them, oh, I noticed this sometimes and this, and that may be, we don't kick ourselves about that, but it'd be nice if we didn't cause that kind of stuff. And if you said, hey, listen, would you rather be seizing all the time or have that? Most people would take that. But if you said, hey, listen, you could have done a slightly less burning lit and got the same result without causing that, well, of course we'd take it, right? And that's really what it comes down to is, our forefathers were just happy the patients didn't die. And then it was that the patients weren't paralyzed. We have to get to the point where we say, well, we're pretty good at not doing that now. So we need to aim for how do we get the best combination of outcome and function given the circumstances. So if it's a GBM in the motor cortex, we're not going to fix the hemiplegia probably, but don't cause a language problem.
All right, well, it's already 6:40. I'm sure Mike has a full day ahead of him in Australia, and Simon and Dan. I'm sure you're eager to find out who wins the election tonight. But anyway, thank you all for your time. I really, really appreciate it. It was a fantastic talk and fantastic cases by Dan and Simon, and I hope everybody stays safe. And if you guys want to know more about Mike's work, he does have another talk coming up, I think, next week with the CNS. Please feel free to tune into that or get in touch with them for more information.
Well, thanks, everyone.
Take care.
Thanks, guys.